So today we will talk about the new drug approval process, and in the next lecture we'll talk about the clinical trial design. Now, the structure above is for a drug called osilodrostat, and according to the website of the FDA, it was the most recently approved drug. It's used for a hormonal excess syndrome called Cushing syndrome. So, in fact, the FDA has a website that provides any newly approved drugs and their indications and the link for the drug. The major process of approval of drug is conducted to make sure it is safe and effective. And many companies, in fact, they spend a lot of money on their products, but few of the party responsible for the approval of the drugs or new drugs is called the Food and Drug Administration, or the FDA. For the FDA, before it came out, there were three, let's say, laws that in fact preceded the FDA. So the first one was the Pure Food and Drug Act, and it came after the publication of an article, it's called an expose, written by Upton Sinclair, that exposed the dirty business of meat packing or storage in the United States. This article was called The Jungle. Now, after that, the Food, Drug and Cosmetic Act 1938 came out as a result of the manufacturing of sulfanilamide and incorporation of a solvent, a dangerous solvent in it. So the diethylene glycol was used to solubilize sulfanilamide, and it has high toxicity. It would be activated by the liver, and it will lead to formation of toxic metabolites that were associated with death. So after the Food, Drug, and Cosmetic Act, in fact, this act required from manufacturers of drugs to disclose or include the ingredients and of the product, the formulation, the assay methods, the manufacturing process, and any experiments that preceded the approval of the drug, like animal or human testing. إداريين. Now there are some subdivisions of the FDA or FDA affiliated centers. One of them is the Center for Food Safety and Applied Nutrition, and this center oversees food and cosmetic or مواد التجميل products. There is the Center for Veterinary Medicine, and it is responsible for animal feed and drugs. يعني veterinary يعني طب بيطري, veterinary medicine طب بيطري. And then the Center for Devices and Radiological Health, and it covers the safety and efficacy of medical devices like drugs used for diagnosis or even treatment or therapy of patients. And then it oversees or supervises the use or approval of radiation emitting devices such as lasers. Those are used to perform surgeries. X-ray systems, those are used to diagnose patients, and also microwave ovens and even cell phones as they emit radiation. In fact, there are some studies that show that drug. So, as we have said, there are FDA equivalents in many other countries. Like, for example, in Europe, there is something called European Medicines Agency. It is more or less responsible for whatever the FDA releases. And even all of those companies, they work in harmony or try to unify their findings or information provided to the patients, or like to be on the same page. They have established something called International Conference on Harmonization, يعني إنه التناغم, المؤتمر العالمي للتناغم, to develop standardized regulatory processes for the major markets and allow results of international clinical trials to be used to support approvals across countries. So this is something that makes data available about clinical trials to everybody that takes the drug, whether they are from Europe or from the. So for the US drug approval process, it is, as we have said, it goes through the CDER. The same rules and regulations apply to the biologics, but for medical devices, the situation is different. They require something called the investigational device exemption approval before you go into FDA approval. Now, for drugs, they first require development and then testing in animals. So this is required before the drug is approved as to be tested in human beings, and then it goes into clinical trial. Now this flow chart shows the events that happened during the approval process. So the first step is the discovery of the drug, and then the drug is or the nucleus or the pharmacophore or the general structure of the drug is selected, and then this undergoes like manipulation or changing to produce something called the lead compound, which is the optimal drug to be effective in whatever like treatment we are using it. Then it goes into preclinical testing, and the preclinical testing period is around like 4 years. It will be tested in vivo, let's say, and then according to its activity in vivo, it could meaning in vivo, like in animals, it undergoes animal testing, and then when they approve it or make sure that it is effective in vivo, so they have to prepare the data required for its clinical testing. So they have to manufacture the drug on a large scale, like in a factory or in large amounts, and then this would be followed by the clinical testing. The clinical testing drugs. The drug discovery is a process where you try to develop a drug. Like previously, serendipity or coincidence played a major role in discovery of new drugs. But nowadays, the discovery of drugs is a process, like a complex process. So we have either the finding of a pathway in a disease, and this requires studying the disease, studying scientific benefit. Now, the drug discovery process, in general, it could, as we have said, you could have either a targeted synthesis, or you could use combinatorial chemistry, or you could use drug modeling for the synthesis of the drug, or you could use a combination of three, and even we have many other. So with the targeted synthesis approach, researchers focus on one step in a disease, such as like cancer, and they pick a process to be targeted by a drug therapy. Like, for example, this is a huge pathway for a major molecule in cancer, which is called epidermal growth factor receptor. So simply, scientists would target either like the AKT or they could target the PI3 kinase pathway. In the combinatorial chemistry approach, scientists use a molecule called the pharmacophore, which is the minimal requirements for a drug to be effective in any certain condition, and they use it as a base for a chemical. So they add multiple functional groups to it and test for their activity. This approach was used to develop statins. Like, for example, the structure to the right, the small structure to the right shows the minimal requirements for the effect of a hypercholesterolemia drug, or a drug that lowers cholesterol levels. And with modifications added to this compound, we produced lots of statins. In the drug modeling approach, computers are used to find drugs that could bind to certain structures, and those certain structures would be enzymes, receptors, transporters. So they try to use computers to calculate which structures could in fact bind or fit into certain binding sites on that structure, and you could calculate the strength of binding of those compounds. So this approach has initial high costs. Like, for example, you have to have the institute or a facility to have computers, and the computers, they need software, and the computers should be of high speed and certain high cost, like graphic cards. Those are initial high costs, but with established institution or facility, you could pursue this work. So the preclinical testing is important for many reasons. Mainly, they rotate around the properties of the lead compound, such as the in vivo activity, the ability to synthesize it in a large scale, and the purity of that compound, the formulation used to prepare it, or the components other than the drug that are available in the formulation of the drug, and animal data to ensure the safety and report the toxicities of the agent. So for some drugs, in fact, preclinical studies were performed outside the United States. So for those people, they could submit their results to the American government or the FDA through pre-IND meetings. IND stands for Investigational New Drug. So probably any data, animal data, clinical data could be submitted to the FDA through the pre-IND meetings. So when preclinical testing is done, the sponsor or the manufacturing company could file for an application called IND, or Investigational New Drug. There are some guidelines that are provided by the FDA for the submission of the IND. The application should include the name of and chemical description of active components, list of active and inactive components, manufacturers of components, method of preparation and dosage form to be administered, preclinical animal data, names and locations of the investigators performing planned clinical trials, data from clinical trials conducted in other countries, like in pre-IND meetings, and study protocols implemented to evaluate the drug. And study protocols include study procedures, informed consents. If you are conducting research in humans, they should agree on conducting the experiments. So they should sign something called informed consent. Data collection and analysis procedures and mechanisms of for subject selection or randomization, follow-up, how to follow up them, and how to monitor safety of the drug. Now, when the IND application is submitted, the application receives a number, and this facilitates the ability of sponsors to track the application and track any correspondence or communication from the sponsors or to sponsors. As we have said, it's the responsibility of the CDER to accept the application and process it, and there would be a 30-day evaluation period. And after that, the FDA will give it an approval or clinical hold. And clinical hold status is put on applications which are not complete, and it is subject for revision. Also, the facility where the clinical data or clinical data will be collected or clinical trials will be conducted should the sponsor should get something from them called IRB, or Institutional Review Board. And it is the IRB is a committee that revises the procedure of the study, and this committee could hold or put the application on hold if there are any of the following violations, like unreasonable or significant risk of illness or injury to trial subjects, insufficient information or procedures to assess and minimize patient's risk, or inadequate qualifications of clinical investigators, or a misleading erroneous with errors, or having error, or incomplete investigator's brochure. And a brochure is a document that contains all relevant information of the drug regarding mechanism of action, activity, effects, side effects, any related information. And also, there is an annual report that is required to be submitted by the sponsors while they are conducting their experiments or clinical trials, and this should be annually submitted to the FDA upon approval of their studies. Now, there is a link for that. As you can see, there is a link for the format of the annual report that should be submitted to the FDA. Now, the clinical investigations, as we have said, those are clinical trials conducted on human beings. And there are 3 phases of clinical trials that are required to be completed before the approval of the drug. And after the approval of the drug, there would be a 4th phase of clinical trials, or it's called the post-marketing clinical trials. Now, there are some differences between the phases of clinical studies. Phase 1 studies, they include a small number of subjects, around 20 to 80 people, and they are healthy, non-diseased patients, except for some other conditions where you have to enroll patients. Like, for example, for HIV patients or AIDS patients or cancer patients, you do not enroll normal subjects in the phase 1 trials, but rather you enroll a small number of patients. And then the starting dose here is not known, or the optimal dose. And in fact, researchers would extrapolate or use the doses that have been effective, in fact, in animal models. So they use 1/10 of the highest no effect dose in animal models. So the highest non-effective dose in animal models per weight is used to extrapolate or guess the dose per weight in human beings. And usually phase 1 subjects, they have around 70 kilogram weight and 30 or they are 30 or 35 years of age. And one of the main purposes of the phase 1 clinical trials is to determine the efficacy and safety of the drug and toxicity. And it is also used to determine the maximum dose tolerated without significant side effects of the drug. And absorption, distribution, metabolism, and elimination or excretion, which are called ADME, or pharmacokinetics of the drug, are determined in normal subjects. I should note that ADME, or those pharmacokinetic parameters, they are not similar in disease patients, and they could be different. Now, in phase 2 clinical trials, there would be an enrollment of patients. Number is higher, and they are suffering from the disease. So they are patients, and side effects are noted in this phase of trials. And then failure is common during that phase 2 study. As side effects, more side effects will show up in that phase of study because we are using a larger number of patients. In phase 3 studies, in fact, those are pre-approval studies. And before going into the phase 3 studies, sponsors or companies should review previous protocols based on study results acquired in phase 1 and phase 2 studies. So like protocol of the study, informed consent, study objectives, people who are included or removed from the study, like inclusion and exclusion criteria, dose regimen, like dose, how many times per day for a certain duration of time. This is the dosing regimen. If monitoring is performed or levels of the drug are measured in the blood, so timing of collection of data is essential for establishment of the levels, or even just to monitor the drug. So those the timings are perfected or modified in protocols of phase 3 study. Duration of treatment, follow-up assessment, many of those are reevaluated. Definitely, when phase 3 studies start, number of patients would be higher, and it would be, the patients would be from different countries or different locations in the country. So it would be a multi-center study. And definitely patients are enrolled, not normal subjects. And relatively, this phase would be the longest in duration. And 95%, in fact, 95% of drugs that enter phase 3 studies would be FDA approved, and those phase 3 studies, they in fact are conducted to fulfill the requirements for the next step, which is the approval of the drug, the new drug approval application or new drug application. When phase 3 studies data are collected, they are submitted to the FDA, and the FDA decides whether to approve it or not. And in case of approval, then the company is required by the FDA to conduct phase 4 studies, and they are mainly aiming to collect more data on the effectiveness and especially the toxicity of the drugs. Now, the new drug application requires the compilation and the collection of preclinical and clinical data and submitting them into the NDA or new drug application request. This is reviewed by the FDA, and the application's primary items include safety and efficacy of the drug treatment, components of drug, description of methods and controls used to manufacturing the active ingredients and drug delivery system and its packaging, and the proposed labeling or leaflet of the drug. Now, the number of or the waiting time for the application to be revised has developed, and 13 months are the average time for wait for the application to be approved. Now, some drugs require faster approval, especially if we're talking like about coronavirus therapies, or this situation is very critical. So 6 months are maximum wait time for a priority drug or a priority review for this drug. So according to the drug that is being approved, we have a designation for whatever drug. So a drug receives a P review designation or priority if it was a new drug modified from a previous drug currently approved in the market in regard to therapeutic effects, safety, side effects. And we have the standard application. And even for a standard, a decision is taken within 60 days of application, whether to approve it or disapprove it, or even approve it with specific conditions. And definitely the decision would be corresponded, or the FDA would communicate with the sponsor or a manufacturing company with the decision and the reason for whatever decision that was made. So for the disapproval could be caused by lack of demonstration of safety and efficacy of a drug, issues with manufacturing, processing procedure, or false or misleading data that are found in the labeling. Now, for the clinical trials, while they are ongoing, there is an option of seeing the status of those clinical trials. Now, those clinical trials are available on a website called clinicaltrials.gov, and this was made in collaboration of
in collaboration with the NIH and the FDA. NIH stands for the National Institute of Health. The website is searchable. You could search for the condition, for the drugs, and it will provide you with the purpose of the study, recruiting status, are they still bringing patients into the clinical trial, or normal patients or normal people, criteria of the participation, meaning the criteria that makes you include a patient or exclude him, which are also referred to as inclusion exclusion criteria, the drug that is being studied, and the location of the clinical trials. So the investigators are mandated to provide the results on the website once the trial is expected, and especially if the results were negative, meaning that adverse effects were noted or there were no significant outcomes of the study, and they should be published on the website and made public. Now, for some drugs, we know that when a drug is FDA approved for the first time, it will carry a brand name. Now, this brand name is protected by the law so that nobody could manufacture the same drug and sell it to the public. This is called patency or patency or براءة الاختراع. Now, the patency will guarantee that the brand would be protected and nobody and the company will get a benefit from selling that drug until the patency ends, and here the generic would be available to be produced by other companies under different trade name. Now, those usually receive an abbreviated new drug application, and the manufacturer of the new trade name, they are required to provide less data for the drug concerning mainly the release of the drug from the formulation or bioequivalence testing. Since the safety and efficacy are already established by the manufacturing company. Now, the manufacturing company or the manufacturer of the new trade should also provide in vivo studies like in animals, but they could be exempted, or they don't have to provide in vivo studies if there is a study that shows a comparison between the formulation of the old branded drug and the new generic drug, and a comparative dissolution, or meaning a formulation and a dissolution test. And it could be delayed if approval of the generic occurs prior to the expiration date of patents or exclusivities of the reference drug product. Now, for some drugs that are produced for critically ill patients, like cancer or AIDS, they could have an accelerated approval, or you could have rapid access, or healthcare workers could have rapid access to those drugs. They could be available during phase 3 studies. And so the data provided on those drugs could be limited. So they receive a special type of approval, like it would be an approval, conditional approval, and for the manufacturers to convert it to an official approval, or a regular approval, they have to provide additional data to convert the status of the drug. So for those diseases like cancer or HIV, a main endpoint for the effectiveness of therapy is the reduction of the death cases or mortality or survival, especially for cancer. So the ultimate endpoint for the results is the mortality, which needs more time to be determined. So instead of that, manufacturers would use surrogate endpoints or secondary, let's say, endpoints that are not directly a measure of mortality, but a direct measure of benefit or effectiveness of therapy for a short period of time. Now, as we have said, so during the phases of clinical trials, you have to recruit patients to be involved in the clinical trials, and this is very hard, especially for those diseases and for an investigational drug to be used in a clinical trial. For the drugs that have received accelerated approval from the period from December 1992 till July 2010, we had 35 new drugs for 47 indications that have been approved or received an accelerated status or accelerated approval status. Only 26 of them, they were converted to a regular approval status. And in fact, some physicians could use some drugs without having an indication for that drug. And this is called an off-label use, meaning that it's not available on the label of the drug. For some diseases which are rare, a rare disease is defined as a disease that is affecting less than 200,000 people in USA. So those drugs are called orphan drugs. Orphan معناها يتيم. And those receive something called expedited review, like a fastened or a fast review process, mainly because the number of patients is not high, and it's hard to recruit patients to those clinical trials. And because of the low number of patients, you would not expect the drug to receive to give back the money that was spent on its development. So the government is encouraging the companies to work on those drugs by the release of a law or an act called Orphan Drug Act. And this act, the 1983 Act, gives those companies that develop orphan drugs tax incentives, and they reduce fees on those drugs, and they could even receive agreements, exclusive agreements with the government to support their research on those orphan drugs. In 2012, only 39 new products or orphan drugs were approved. Now, orphan drugs could be found on the website of the FDA. You could search those orphan drugs. So I performed the search from January 1st, 2019 till March 14th, 2020. And in fact, 25 drugs have been approved in those, like, let's say 1 year and a quarter of a year. Now, for the approval process of the over-the-counter drug or OTC, or those are drugs that are sold without a prescription. Their process of application, new drug application, is different. The application is first submitted and it reaches an advisory committee, which revises data provided by the manufacturing company and any publication related to that drug. Then that advisory committee, they submit their their revision to the FDA, and the FDA summarizes the data in a federal register, and they wait for any comments about their summary from the manufacturing company, and then the FDA publishes a tentative monograph or a preliminary one, and then they finalize the publication. The monograph established conditions under which the OTC drugs are recognized as safe and effective and are not misbranded. And any additional changes to the indications could be modified by the manufacturing company by providing data to revise the monograph, or they could submit an NDA if there is a new indication for that OTC drug.